Hi, my name is Kira Nakamura, and I'm from San Francisco, and my project is focused on the migration of interneurons in the postnatal piglet brain. So interneurons are a subpopulation of neurons that are really important for brain development. And I used piglets as an animal model because they have gyrification, which are these folds in the brain similar to what humans have. And they also have similar subventricular zones, which is where this structure called the ganglionic eminence is, and that's where interneurons originate. And cells migrate from the ganglionic eminence to the arc, which is this region where there's a high population of young migrating interneurons. And from here, interneurons begin to migrate outwards. So these are maps of slices of the brains where the interneurons are each marked with a green dot. And then figure 3 is a zoomed in image of some of these populations of migrating neurons in various cortical regions. We can also see that the morphologies of migrating interneurons are a lot more complex, and they start to mature. I was also interested in tracking the origin of interneurons from the arc, because we know they come from the ganglionic eminence, but that's a region that has 3 distinct parts. So there's the MGE, the medial ganglionic eminence, the CGE, the caudal ganglionic eminence, and the LGE, the lateral ganglionic eminence. And we know that most cells come from the MGE and CGE, but by using stains specific to those regions, we saw that there's a greater population of CGE-derived cells in the postnatal piglet brain. So from all of this, I was able to find that interneurons are migrating after birth, and therefore brains are still susceptible to environmental influences. And by looking at more extensive mapping of interneurons throughout development and the regulation of cell development, we can understand how interneuron migration can contribute to neurodevelopmental disorders like autism or epilepsy.